So today we're going to be talking about a little bit of a continuation from last time, talking about if plant cells are in a box, how do they ever expand and grow? So last time we talked about how they can elasticize or stabilize their walls by modifying pectin chains. And we had talked a little bit last time too about how just making the wall stretchier will cause the cell to expand if water is trying to move into the cell. So when the wall is rigid, as depicted here, there is water coming in, it is pushing on the wall, and the wall is pushing back. I think today is going to be a day when I'll probably need the screen up all day. So in this picture, the wall is pushing back at a level that is equal to the water pressure coming in, if that makes sense. And when we say in this unit turgor pressure, the turgor pressure is like water pressure. It's just called turgor pressure when it's inside the cell. So if a rigid wall pushes with an equivalent amount of force, a loosened or elasticized wall will still be pushing, but it's pushing with less force. So these pictures are proceeding in order. So a cell without loosening its wall will maintain a certain size. As soon as the wall is loosened, the force that it can exert is less. And the blue arrows overpower the red arrows. The turgor pressure is greater than the wall resistance, and the cell will expand. The turgor pressure is pushing out against the loosened wall. To get to the point to restabilize the wall of the expanded cell, the wall needs to be made rigid again. One of the focuses, this will be true for every year, not just every year that I teach this class. One of the focuses on the exam is how do changes in pectin methylation or pectin cross-bridging affect how big a plant can grow, and what are different factors that involve that. So we'll practice some of those questions on Wednesday. So at this point, the cell has expanded because water has come in. Water has come in because the wall is not holding it back. The wall resolidifies. And this is a better pictorial review of what we talked about last time. Just got pictures with it. Questions about this? Yeah.
Are the lines that are drawn, are those indicating how water gets through the wall? Or
I am not sure about that. No, they are not.
Okay.
I had to think about that for a minute. The wall is not impervious to water one bit.
Okay.
So if you think about go back in your slides to where we had the crisscross of cellulose and glycans and pectins, there's plenty of space in there for water to drift through.
Okay.
It's so small. So these are not like aquaporins. These are just some random white lines, I guess. It's a good question. Okay. So this situation of plants taking on water to provide turgor pressure to either plump up a cell or to expand the cell, it depends on impetus or the ability of water to go into the cell. Water has to want to go into the cell. And we talked about last time that the plant had better be in a hypotonic environment to make that happen. Where does water go into a plant? In what part? How do plants get water in the first place?
Their roots.
Through the roots, yeah. So plants will be taking in water through the roots if they have true roots. Assuming they have true roots, higher plants. And the solution in the soil that they're pulling in needs to be hypotonic compared to what's inside the plant. Come back to that in a minute. But that's what some of these pictures are going to be talking about. So if water is going to move into the plant cell, or is going to move into the vacuole of the plant cell, it has to be coming from a hypotonic solution. Hypotonic does not mean anything on its own. It just means that the solution outside this lovely plant needs to have less solute or be hypotonic there compared to there being more solute inside the cells. And we're going to do calculations with this in a minute. The way that plants get water to come into them is to make solutes come into them. They're making themselves hypertonic compared to the outside solution. And the way that they do this is by accumulating solutes, dissolved particles, in the vacuole for the most part. This is also one reason why vacuoles are really important for plants. So the figures that I use in here, they have this certain type of schematic that they show these characteristics. So you've got a compartment, which in this case is the vacuole, and then you've got the outside conditions. There are some new words that I want to define that relate to how plants take up water or not. And those are osmotic potential is one. And I'm going to leave space to define that. There's another one called water potential. And the third one is turgor pressure. Turgor pressure, we already talked about. And that's the physical force of water pushing on the walls. So if your faucet won't run because you have no water pressure, there's no water pushing through the pipes to bring water up through the faucet. So this one, that one's easy to understand. Like, how hard is water pushing against the wall? That's turgor pressure. Osmotic potential and water potential, we're going to define in just a minute. Also, these pictures are indicating the direction of the gradient. So these triangles that the book uses, they're showing high to low. So where the arrow is, or where the triangle is fat, that's high part of the gradient, and it's directed toward the low part of the gradient. That's what those mean. So if a plant is going to be able to take in water, it has to accumulate solute in the vacuole. This picture is about recalling that a plasma membrane of a plant cell lets water through fine, but not most other solutes. Those of you that are in cell, like, same concept we're going to talk about Tuesday in cell. So the cell wall, is not really a molecular barrier. It's got pretty big spaces in it, and molecular is what I'm trying to write here. It's not a big-time molecular barrier, but the plasma membrane lets water come through, oxygen, and CO2. Those are the only things that can pass through just the plain cell membrane of a plant cell. Water can go through, most other solutes do not. This picture is showing two sides of a membrane, and we're going to assume that this is the plasma membrane. And we've got blue dots, which are water. Blue marker, got a green marker. Blue dots, which are water, that can go through the plasma membrane, and red dots are some other solute. And I'm sure you remember from Bio 1, water moves down its concentration gradient to try and balance solute levels. You remember that? Okay, do you remember this? If you were going to call one side of this, if you were going to name these two sides hypotonic and hypertonic, which is which?
Hypertonic would be the right and hypotonic would be the left.
That is correct. Hypertonic is the right because it's got more solute than the other side. And those two terms have to be relative to each other. So this side, at this moment in time, is hypertonic. This side is hypotonic. The way that I think about this is by putting actual numbers on it. So let's say if this side, hypotonic, is 93% water and 7% solute. This side is 84% water and 16% solute. And I'm just making up those numbers. This side has more solute. This side has less solute. In plant biology, they don't tend to use hypo and hypertonic. They talk about something called water potential. Water potential is related to the percent water. That's what I think of, how I think about it. Water potential is the concentration of water. A higher water potential has a higher percent of water. You can also think of it as dilutedness. So where you have more water, that's an area of higher water potential than an area of lower water potential. And if I were to draw one of those gradient arrows for the direction that water moves, it would be like this. That's the direction of the water potential. So water does in fact move that way through a membrane. The osmotic potential is related to the concentration of solutes. Not just one solute, but all solutes. That's osmotic potential. And the osmotic potential is normally going to be the opposite direction of the water potential. So if a solute could move, which direction would it move? They would like to move from here to here and spread out by diffusion, but the membrane blocks them. But if they were able to move, they would be moving from an area of high osmotic potential over here to an area of lower osmotic potential over here. Does that make sense? Osmotic potential is soluteness. Water potential is dilutedness. I don't care how you remember it, but you need to be able to label where is high water potential, low water potential, which side is greater osmotic potential, and so on. This slide's really important. Know these definitions. I'm going to erase and move on to the next. water will move from its own high concentration to its own low concentration through a membrane up until the point that it's stopped by a cell wall. So going back to this kind of picture, the reason that water would try to come in here is that there's low water potential inside compared to outside. So water will be attempting to come in, raising the turgor pressure as much as it can until the wall balances that out. Yes?
So is the water, so water is not moving into the cell at the point of that first picture?
It is moving in and out of the cell, but the cell's volume is constricted by the wall force.
So water pressure on the outside of the wall forces it to become more flexible?
Water is coming like up and through the wall, can come through the membrane, but it's like no more water overall can fit in there because the wall is binding it. I don't think, is that answering? It's like if there was something in here like donuts,
Yeah.
and people wanted to come in and have donuts, we've got the impetus to bring donuts in here. We have a high donut potential. We have a low eater potential. But we could not fit endless numbers of people in here. So in this case, water doesn't reach equilibrium. This cell will not expand infinitely because the wall is holding it in. If you took this cell and you made a protoplast out of it and took the wall away and it became round, it would be easy to just burst it by letting water come in. Is that helping?
It does. I'm just confused, like what is, like, I mean, I know there's not like a clear signal or something like that that's telling it to like loosen.
There is actually. I just don't get into that in this class.
Okay.
There are a lot of signals and hormones are involved, and we are going to look at a signal that comes into a guard cell and changes something to let more water come in. There are signals.
It'll probably make more sense when we get to
Yeah,
that. Okay.
probably. But most cells in a plant are always sitting in this scenario where more water would like to come in, but the wall is holding it back.
Okay.
And in fact, they need to maintain that situation so that they stand up and don't wilt. Good questions. Okay, so water wants to come in. Water wants to come into plant cells because they are maintaining generally higher solute concentrations or higher osmotic potential inside their cells than the water in the ground. Water can go across membranes. Water can also go through proteins called aquaporins. Aquaporins. I think these are in kidney tubules too, right? Do I remember that right? I think they are. So if water cannot get in fast enough through the membrane, certain cells can create aquaporin channels that are the perfect size for water to rush through even faster. That's more of just a fact slide. Okay, so the plant needs to maintain high osmotic potential so that it can take up water, plump itself up. To do that, it needs to keep solutes in the vacuole. How does it get solutes in the vacuole? It spends energy to do this. This is so important for the plant. It spends energy to push solutes into the vacuole. If you've had mammalian phys or talked about sodium-potassium pump, sort of the plant equivalent of that is a proton pump. So this boring blue box on these slides, this is a proton pump. And these run constantly in plants. plants use proton pumps to maintain gradients across the membrane. They're always running. They can be boosted in their speed. They can be suppressed in their speed, but you can consider them as always running. Okay, so here's our gray plasma membrane. Here's the cytoplasm. Here's the outside of the cell. These proton pumps constantly run. They constantly spend ATP to push protons out of the cytoplasm. They push protons out of the cytoplasm. They can be upregulated by plant hormones like auxin, which I'm not getting into today. So protons are pushed out outside of the cell, away from the cytoplasm. And that means that the outside of a plant cell is generally more acidic than the inside. So we've got a proton gradient created by this that is directed in that fashion. Since there's more protons out here, it's more acidic, it's lower pH. Don't mix that up. It's lower pH, more protons. Those are running all the time. The reason that that's so important to run all the time is that plants use the proton gradient as a source of energy to move other substances. Okay, so pretend that, all right, we just looked at that part. Protons are getting pushed outside. They form a gradient. pH out here is lower than pH in here. And look what type of things will ride back in on a proton wave, so to speak. This would be cotransport. Cotransport. Potassium re-enters the cell with protons. So does phosphate, nitrate, sulfate. This is the odd one out. A lot of nutrients that plants need, which are also solutes, ride into the plant cell going down the proton gradient. Again, those of you that are in cell, we look at the animal systems of this on Tuesday. It's a really good coincidence. So you need the proton gradient to pull in solutes into a plant cell. This fact is unrelated. This is unrelated to protons, but it's just saying that there are channels in the plasma membrane that let potassium flow in and calcium flow in. That movement is not driven by protons. So the more protons out here, the more solutes in here. Having that protein gradient allows the accumulation of solutes. And lets the plant cell have high osmotic potential. And lets the plant cell pull in water. Okay, chemistry question. Why does the plant cell bother to do this? Why wouldn't it just like pump protons in and that's a solute? That would give you osmotic potential.
Because you're not evening everything out.
You're not evening everything out?
Yeah.
How so?
Like balancing everything out.
You're not balancing everything out. I mean, why have these solutes go in and protons go out? It seems like a lot of runaround.
Is it, I mean, most of those ions are negatively charged and the proton is positively charged. Does it have to do with equaling charges in and outside of the cell, or creating like a negative potential in the inside of the cell.
Some of that is related, like plant cells do have an electrical charge across the membrane. Some of it has to do with that. Some of it has to do with, these are things that the plant cell needs anyway. They need those in there anyway. Some of it has to do with, these as a set are bigger than these as a set. And they will provide as a set higher osmolarity and pull water better than just little protons. Several reasons.
I guess also, like you said, like since it needs those anyway,
Mhm.
if it were to have, it would probably have to have like a pump for each one of those, and each one would probably have to use energy too.
Yeah, these are not in essence pumps, they're facilitated, they're kind of like facilitated diffusion channels. They're using a gradient to bring these in. But overall, that's probably energetically cheaper, like you said, than pumping every one of these things in. So this system is hugely important for plants, and it's something very different from what we do. Again, those of you in cell, we've got sodium-potassium pump on Tuesday. This is what plants are doing instead. A lot of the same concepts. This is also, the proton pump is the major pump for bacteria as well, which is interesting. But all of this coming in depends on protons going out, and the plant spends ATP to do that. Okay, now we look at this figure. So we have been looking at proton pumps that are oriented across the plasma membrane. Just the regular plasma membrane in a plant cell. Now we're looking at proton pumps that are going across the tonoplast. So remember, tonoplast is the membrane around the vacuole. So we've got that membrane. Out here we've got the plasma membrane. Which direction was the proton pump pushing protons out here?
Outside.
Out. It's nice that you didn't say to the left.
Always.
What if the proton pump was over here? What direction is it pumping protons?
To the right.
To the right. Or out is the correct answer. So, out. That's important because the proton pumps in the tonoplast are also pumping protons out of the cytoplasm. They're not working against each other. So they have the same general function, the same general shape, but no matter where they are, they're pumping protons out of the cytoplasm. Don't get mixed up left and right if I change, like the figure is a little bit different on the exam. I did that one year. It depends on how I'm feeling. I'm going to make this a mirror image this year. It depends. Okay, so protons are coming in this way into the vacuole. The pH of the vacuole is this. What's the pH going to be out here?
Lower, higher.
It's going to be higher, but the amount of protons is lower. Protons are moving out of the cytoplasm, so this pH is going to be greater than pH 5.5. The vacuole is accumulating protons. It's also accumulating other ions. But they are not riding a proton wave. They're being pumped in as well. So it's accumulating nitrate, chloride, and sugars. Need a new marker. It's accumulating sugars. So in the vacuole, water comes into the vacuole based on the movement of these things. And the vacuole swells up with water. The vacuole is a place where there can be aquaporins in the tonoplast membrane. So you can have proton pumps in the plasma membrane, you can have proton pumps in the tonoplast as well. There was a blank here to fill in. So the vacuole is accumulating solutes. The vacuole does not have the channels. This is important. The vacuole doesn't have these. The vacuole doesn't have these. So the plant cell is accumulating those solutes in the cytoplasm, and it's also pushing these particular solutes into the vacuole. So overall, the force of water is still coming inward. So we've got more solutes here, there's more osmosis into the vacuole, and there is higher turgor pressure. There is higher osmotic potential. There has to be if water is moving this way. What about the water potential inside the vacuole? Well, it says it on there. There is lower water potential. Lower water potential. Does that seem okay? Is that making sense? You know this rule, water follows salt? You ever heard that? One day Noah came with me to a lecture in A&P 2, and it was on a day about osmosis, and he was 4, and he remembered water follows salt. And he doesn't know what it means, but that's going to help him someday. Water follows salt, or water follows solute. Super helpful to remember. In kidneys, in blood cells, in bacteria, in plants. So making solutes build up and pull water, plants use this to do some pretty interesting things. So when a Venus flytrap hinge snaps shut, the reason it does that is that back in this region behind the fly, there are vacuoles that are filled up with water, and they are holding the sides open. When they get a signal from this fly stepping on 2 individual hairs within a certain time period, and you're going to read about that in a few weeks, they get a signal to make water go out of the vacuole, and it does, and it snaps on the fly. They're using water flow to provide that fast movement. Pretty much every fast movement that a plant makes is this water flow effect. Water comes in, water flows out. That's the main way that plants do a fast movement, which is cool. This is sort of related to what Sunny was asking. Why would water not keep coming in? So if we take a plant cell and we take away the wall and we degrade it chemically, we have a protoplast. The way to get solutes to come into a protoplast is by the action of a proton pump. And that would be the same proton pump that we looked at before. It's still there, whether the wall is there or not. And if the proton pumps keep on running, the plant will keep on accumulating solutes. If you're going to do that to a plant, you have to be really careful that out here, let's say, outside the protoplast, the solution where you're maintaining these, this can't be too hypotonic. It can't be too hypotonic, otherwise water would rush in and just eventually burst this. So the solute concentration out here has got to be balanced. Like, I don't know why anyone would do this, but I used to work in a lab that did protoplast solutions. Actually, it was callus, but you know, if you want to ruin your neighbor's work, just go put a bunch of extra water into their tissue culture. They're using protoplast, everything will die. Don't do that. Probably no one has ever done that. It's a bizarre, dorky kind of sabotage. This I'm not going to go through, other than to just say the force of water coming in. The force of water coming in is not equaling the osmotic potential that is in here because of the wall. Because of the wall. So this has high osmotic potential, lower water potential than out here, and water cannot come into equilibrium because the wall is preventing any more from coming in, so to speak. This is repetitive. I'm going to skip over this until next Wednesday because I want to get on and talk about stomates and how they are using water flow to open and close. So remember that stomates or stomata are these openings on the lower surface of leaves usually, and they are allowing CO2 in. So these are the net flow of materials through stomates. CO2 is going in, oxygen is generally coming out because plants are making oxygen during photosynthesis. So the net flow of oxygen is out, and the net flow of water is also out. That doesn't mean that some water can't go in, but overall there's more coming out. Like when this class is over, there will be a net flow of students out of this room, even if one person is trying to come in, net flow out. Plants have to have CO2 to do photosynthesis. Thus, they have to have stomates open at least sometime to let it in. Why can't CO2 just go straight through the leaf? It goes straight through membranes. It goes straight through walls.
Cuticle.
The cuticle. So all over the surface of the plant, there's waxy cuticle that keeps water inside the plant, but it won't let CO2 through. So this is, I don't say this is how plants breathe, but kind of. Have you studied stomates before, beyond what I just said? Okay, I wouldn't expect you to really. Just going to see. So stomates are openings. They're surrounded by two guard cells. And these cells are specialized. And they're shown here like kidney bean shape. Those are the guard cells. Please note that guard cells have no plasmodesmata. They are not connected to their neighbors. They have separate cytoplasm from their neighbors. They also have radial arrangement of their cellulose bands. So the blue here, you can imagine as cellulose bands. And the cell wall that is next to the stomatal opening is thicker than the cell wall everywhere else. Have you ever had a balloon that, like you blew it up and you had fun, and you came back a few days later and it's warped? And like part of it's more constricted than the rest. It's making the balloon form a weird shape. That's what this is like. So it's got an inflexible wall next to the opening. The rest of the wall is pretty much acting normally, but it's got radial cellulose bands. This arrangement of guard cells is closed. Relatively closed. So we're looking at the stomatal opening is closed. They never totally close. They don't ever completely shut, but it can get wider or narrower. And in this version, the guard cells, or GC, contain less volume. They contain less volume. So they have lower solute content, lower turgor pressure, less water is trying to come in. When stomates open, all of that reverses. So we have stomata open or more open. The guard cells are containing more solute, there's higher turgor pressure. This cell has grown bigger, but when it does so, the way the wall is arranged, it opens. Sometimes that's confusing for students. They think, oh, well, if the guard cells are taking on water, that's going to close the stomate. When the guard cells take on water, they become more curved, and that actually opens the stomate. Maybe make a note of that. A guard cell with more water gives a stomate that is more open. So when guard cells take on water, they open the stomate. And for that to happen, it depends on the cell walls having this particular arrangement and on these cells not sharing cytoplasm with their neighbors. So on a regular day, in a regular situation for a plant, they need to have their stomates open. Why do plants need CO2 in the day?
What is it?
They're using sunlight in the day to do photosynthesis and to fix CO2. So the normal situation is plants have stomates open in the day. And they have a cool system for opening stomates in the day. They have proton pumps that are upregulated by blue light. Blue light, like within daylight, it's a mixture of colors. The blue wavelength of light upregulates the proton pump. It makes it more active. That's an excellent system. Lights are on, proton pump works faster, and notice we've now got a guard cell. We're looking at a guard cell now. This is the plasma membrane. Protons are being pumped out outside the cell. When they come back in, they could help carry solutes and help this cell accumulate solutes. And when the solutes accumulate, water rushes in too. There is also a separately controlled potassium channel pushing potassium into the cell. This is not very related to the proton thing. It has separate control, but it is pushing solutes in. So protons coming out, carry solutes in, water follows, the guard cell will take on water and it will open the stomatal pore. These are always in twos, so they ought to be working symmetrically, you would think. They're supposed to. I'm going to go on to the opposite situation so that you've heard of this before we work problems on Wednesday. Okay. First, make a note that at night the proton pump is going to go back to like baseline level of operation. It doesn't completely stop, but it goes down to a lower level of catalysis, let's say. Light upregulates it. So no light, less protons pumped out, fewer protons pumped out, fewer solutes come in, less water comes in. And that would cause, stomates to return to this configuration. So this ought to be at night, this ought to be in the day. Maybe make a note of that. And we would also want stomates to be closed if it's dry. So plants, they're having water come out of the stomates, they're having CO2 go in. If they really need to conserve water, they will want to close stomates even in the daytime. Okay, so now we're looking at drought situation. This chemical, ABA, it stands for abscisic acid, and I'm going to write that up here, abscisic acid, ABA. It's a plant hormone. And plant hormones individually can have multiple functions. One of the functions of ABA is that it's produced during drought. And when it is produced, it does two different things that relate to stomates. So the first thing that it does is it downregulates potassium influx. So having this negative arrow come over to the influx channel, it means ABA is repressing or downregulating the action of that. And it also activates or upregulates solute efflux channels. So these are channels that can open and let solutes out. These channels are present in this situation, but they're not shown, and they're closed more often. So they would be here too, but in a mostly off configuration. The presence of ABA opens those up so the solutes that were inside the guard cell can come back out. So the combination of those actions, it keeps more potassium out, it lets extra potassium leave the cell, it lets other solutes also leave the cell. The solutes are leaving the guard cell, water follows. The guard cell deflates, the stomate closes. It's controlled by a drought hormone. So that gives two scenarios. What I want you to do, I'm like determined to show you that quiz, which is about another area of class. What I want you to do before Wednesday is to go back to these pictures, these two pictures, and label them for the directions of the osmotic potential, the directions of the water potential, and I mean which way is the gradient pointed, like which side is high, which side is low. The pH of each side of the membrane. Something else. Just those three will be fine. One other fact that you need to know about that is that you can consider that it never goes to equilibrium for water to come in. So no matter how much water has come in to an open guard cell, there's still more that would like to come in, but the wall is holding it back. So you need to assume that the wall is always rigid. Guard cells don't really, this may be an overstatement, but guard cells don't mess with their pectin bridging. They want always to have rigid walls and not elastify them. So on Wednesday, that's the next day we're going to have lecture. We're going to practice problems with this scenario and also go back and talk about these mutant phenotypes here.